This section of notes, we will be reviewing RNA and protein synthesis, beginning with the difference between DNA and RNA, and also learning about transcription. We will save translation for a later video. So go ahead and if you have not already, download the guided notes from Google Classroom or grab a paper copy from up front. Let's begin with the learning objectives. So in this section, you are going to first be able to describe how RNA is different from DNA, and there are some very key differences between the two molecules. Next, we are going to be able to explain how the cell makes RNA, which is the process of transcription. And then in the next video, we will learn about 3 and 4, which is explaining the genetic code and how proteins are assembled from the RNA that is made. So we're really going to focus on these first 2 bullets here for this video lesson. Let's start with a few important facts about RNA. So RNA is known as the messenger. It is a single strand, whereas DNA is a double strand. So there's a difference right there. So I'm going to write all of the DNA things in blue here. RNA is made of ribose sugar, whereas DNA is made of deoxyribose. Remember the D in DNA, so that means that the R in RNA stands for ribose. And RNA does not contain thymine like DNA does. Instead, RNA contains uracil. So it still follows the same base pair rule. But instead of apples in the tree, right, like it is in DNA, apples are going to be under the tree in RNA. So apples are under, still cars are in the garage. So if you have a DNA sequence of A, T, A, G, C, G, we can use our base pair rules to write it out as U, A, U, C, G, C. So still the same rules, except now it's A to U, C to G. If there's a U, a T, that means in DNA we're going to put an A in the RNA. And if there's a G, then we're going to put a C. We'll have some more practice on that another time. So what does RNA actually do? That's kind of the basic structure of it, but what does it do? RNA, known as the messenger molecule, because it carries the message, which are the instructions for making proteins. It carries that message from the DNA inside the nucleus to the ribosomes where the proteins are going to be made. DNA on its own can't leave the nucleus. It's just too big. So this tiny RNA molecule, see how it looks a lot smaller than the DNA that we made in class? This tiny RNA molecule's job is to get the important information from the DNA strand and carry that important message out of the cell, or out of the nucleus, and to the ribosomes in the cell where the proteins are going to be made. And there are 3 types of RNA that accomplish this process. The main type of RNA, that's the real star of the show, is called mRNA. So mRNA is our messenger RNA. That's the one that we think of when we think of making RNA from DNA. It's also made of nucleotides, just like DNA, except that this RNA is made of RNA nucleotides, so A, U, C, and G. And it uses the DNA template to make it. So the way we just practiced that, if you had a sequence of DNA that was A, C, G, G, C, A, T, then our sequence of RNA, using this DNA template, hold on, let me label that. So this is our DNA template. So our sequence of RNA from that would be, great job if you think of it ahead of time, is U, G, C, C, G, U, A. All right. So this RNA is also found in the nucleus because it's being made by the DNA that's in the nucleus. So it's in the nucleus to start, but it will travel out of the nucleus to the ribosomes to make the protein. So that is the job or the function of mRNA, is to carry the message to the ribosomes where the proteins will be made. But this RNA does not work all by itself. There are two other types of RNA that are going to help to accomplish the job of turning the message from DNA into a protein. So the second one is ribosomal RNA. And just like the name suggests, this RNA is the one that's found in the ribosomes. So ribosomal RNA is also made of RNA nucleotides, but its job is to make up the structure of the ribosomes. So this ribosome molecule right here is made of ribosomal RNA, that's what these little strands are, as well as proteins. And this structure is going to be responsible for taking the messenger RNA from the top and turning it into our proteins, which make up most of our cell structure. So this ribosome is super important, and it cannot function without the ribosomal RNA that creates its structure. So there's ribosomal RNA, is pretty straightforward. And then the last one is transfer RNA, or tRNA. And I think tRNA is kind of cool because if you look at the molecule itself in this picture right here, it actually kind of looks like the letter T, right? So this is the actual shape of the tRNA molecule. So how cool is that this tRNA molecule looks like the letter T? But the T doesn't stand for the shape of it, it actually stands for the word transfer. So transfer RNA, again, made up of RNA nucleotides. We realized it, they're all three of them made up of RNA nucleotides. But transfer RNA is going to be found in the cytoplasm and at the ribosomes. So transverse RNA's job is to take the amino acid, which is right up here, I'm going to circle it in red. This is the amino acid, this is the building block for the protein, and it's going to transfer it to the ribosome during protein construction. So this tRNA molecule is responsible for finding those little amino acids floating around the cytoplasm, picks them up, and brings them over to the ribosome so that the protein can be made. It's kind of like a taxi cab for the building blocks of the proteins. So if you want to think tRNA, think taxi for transferring those amino acids. And just a reminder down here that the amino acids are the building blocks, the monomers for proteins. We'll talk more about that in our next section. Another thing, before I move on to the next slide, another thing that's pretty cool too about tRNA is that it has what's called the anticodon here. So tRNA does have RNA nucleotides attached to it down at the bottom, and these three specific RNA nucleotides are going to come into play when we talk about translation, the last phase of protein synthesis. So just keep that in mind, that they do have, even though the structure looks a little different, they do have these RNA nucleotides, or like kind of like a message or a code attached to the bottom of them. So let's move off the type of RNA and let's talk about overall the big picture. So I've mentioned it a few times in this section so far, but this big picture is that the message in our cells, the instructions for our cell processes, are included in our DNA molecules. And that DNA is found inside of the nucleus. But the problem is that DNA, which is a big double-stranded molecule that's huge, you guys saw when we were building our DNA strands in class, that DNA can be massive. And it cannot leave the nucleus. It is too big to get out of the small nuclear pores in the nuclear envelope. Here's the little pores I'm drawing on. And it's actually a good thing. You don't want DNA to leave the nucleus because it's safe and protected inside of the nucleus. And if DNA is damaged, that can cause mutations. So we don't want our DNA to be damaged. But we do need to get the message out of the nucleus into the ribosomes where the protein are going to be made. So that is why RNA is so important, because it takes the message from DNA and turns it into a protein. It's like the middleman. And this protein, ultimately, which is made from the amino acid chains, is what is going to build up our cells. It's going to tell our cells what to do. Basically, everything that is part of your body is made of proteins. So that is why proteins are so important. So let's take a closer look at this first step right here. Step number 1 is how are we going to make RNA from DNA? And that's the step of transcription, also called RNA synthesis. So the problem, like I said, is that DNA is too big to leave the nucleus. That is a major problem. And the solution to that problem is to make a section of RNA using DNA as a template. And this occurs inside of the nucleus, where the DNA is existing. Enzymes are responsible for this, just like the enzymes involved in DNA replication, except this enzyme is called RNA polymerase. Not DNA polymerase. Don't get those confused. DNA polymerase builds DNA. RNA polymerase builds RNA. Okay? So this enzyme is the enzyme that is responsible for making the new RNA molecule right here using the DNA template. But after it has made the sequence, it needs to do a little bit of editing, because there's a lot of DNA that we have inside our cells that doesn't actually mean anything. It's just like a placeholder. It doesn't code for any specific traits. So before the RNA leaves the cell or leaves the nucleus, it needs to get a little bit edited. We got to cut out the extra parts that we don't want, like the run-on sentences in a paragraph. So that DNA in the gene is not needed to produce a protein. And those areas are called introns. So the areas that are not needed are called introns. The ones that are important, that code for proteins, are called exons. So exons are important. So when RNA is edited, the introns are cut out. You can see in this picture, we cut out the introns. And then the exons get stitched together. So they get pulled together to form the mRNA strand. There's also a little cap and a tail added on at the ends, because just like we talked about the telomeres in DNA replication to protect the ends of the chromosomes, this cap and this tail protects the ends of the RNA so that the important information, the exons in the middle, are protected from accidental shortening as the cell goes about its everyday activities. So I've got a little bit of a practice. Oh, I don't want to give you the answers to the practice. Hold on. Oh no. Let me hide them real quick. Hopefully you didn't save it. So I want you to take a chance and to try this practice yourself. So use the base pair rules, A to U, C to G. Let me write those in again. So A to U, C to G, G goes to C, and T would become A. So use those base pair rules to turn these template DNA strands into the messenger RNA. Pause the video here, try it on your own, and then press play again, because I will reveal the answer. So pause now. And I will reveal our answer here. So hopefully you got them right. So the first one would be UAGCAU. The next one is AUUCCG. And the third one is GGCUAA. If you got them right, well done. All right, so the last thing is for you to submit that section of notes. So submit on Google Classroom this section of notes for your classwork grade. Submit on Classroom. And then you may begin your homework, which is reviewing what we just talked about in the notes through an interactivity on Savas. Thank you for watching.